So first, I'd like to thank the organizers for the opportunity to present here. I apologize for not being able to come in person. Let's take it away. So today I'm going to talk about actually two subjects: targeted RNA editing as a potential treatment for specific mutations of STXBP1-RD. And also I'm going to talk about our model of iPS-derived motor neurons. I'd like to say that this project started with Lior Dor, which is the father of Ran, a patient with STXBP1, who came to do a postdoc in my lab a few years ago. He wasn't working on STXBP1, but he's actually the one that conceived this work. Everything that I'll show you today was done by Kfir Warshawski. And since I met some of you in the Heidelberg meeting a few months ago, we added Dr. Dan Bar Yaakov, which is another supervisor of Kfir Warshawski, which is an expert in RNA editing. So when I teach molecular biology courses, I talk about post-transcriptional mRNA modifications, and I'm sure that everyone is aware of 5' capping, splicing, and polyadenylation, but other post-transcriptional modifications exist, including RNA editing. So RNA editing means that the mRNA goes through specific bases editing using a specific set of enzymes. These enzymes are called ADARs, adenosine deaminase acting on RNA. This is actually a family of enzymes. There are 3 of these enzymes. ADAR1 and ADAR2 are the best studied one. And these enzymes can do 2 things. They can bind double-stranded RNA. And once they bind this double-stranded RNA, they can go on adenosine bases and do this deamination by removing this amine group, turning them into inosine, which is not a common nucleotide in our DNA or RNA. But inosine forms oxygen bonds with cytosine. So it is read by the cellular machinery, and specifically the ribosome, as a G. And if we look at the codons and the applications that such modifications could have, so it could change the specific codon, it could turn a stop codon into a different codon, and it could affect the expression of the RNA by modifying splice sites, changing the RNA structure and stability, and eventually forming a change of expression. RNA editing has this very nice story in which GluA2, which is a component of the AMPA receptor, is going endogenously through full editing, which prevents the AMPA receptor to being permeable to calcium ions. When this doesn't happen, calcium can go through AMPA receptors, and the ADAR2 knockout mice is actually lethal. And it has been shown very elegantly that by editing the gene GluA2 to already have this modification at the DNA level, they can rescue this lethality in the ADAR knockout. What it also teaches us is that ADARs are abundant in the brain, and specifically there is ADAR editing within synapses. The clinical approach would be to design a guide just like you would do for a CRISPR system, and then when this guide forms this mismatch here, the ADAR could bind this double-stranded RNA and modify A to I. And so this would be suitable for specific mutations. One example is the example that I'm showing here, R46H, which is a G to A mutation. So this codon here, CGC, which is the healthy codon, is changed into CAC. And using this approach, we could change the A back into I, so it would be read as a G, and possibly affect this mRNA to change the protein that comes from it. And of course, this approach cannot be applied to all of the mutations, but from a search that Lior Dor did at ClinVar, he found 55 STXBP1 variants to which this could possibly or potentially be applied. So to recruit the ADAR enzymes, there are a couple of methods that have been developed back in 2019. One of them is called Restore. The other one is Leaper. With the Restore, you have an ADAR recruiting domain, which is a double-strand RNA that is fused to a specificity to a specificity domain that would take this construct or this guide into the target RNA. The other option is just to design guides that are longer, and they also form this mismatch, and both of these approaches has been shown to induce RNA editing. And one should ask why edit RNA and not DNA, since CRISPR is becoming used in specific genetic diseases. So if we compare both, as a molecular tool for DNA editing, we need exogenous enzymes such as CRISPR, whether for RNA editing, we have endogenous ADAR that is already expressed in neurons and in synapses. The molecular compatibility of DNA editing is unlimited, whether here for RNA editing, you can only use G2A mutations. This is what would apply here. Changing at the DNA levels are permanent and irreversible. Whether at the RNA, it is reversible. So if there are some toxic effects, it could be theoretically withdrawn because it's at the RNA, and RNA is an unstable molecule or has obviously a shorter lifespan than DNA. Risk of target mutations exists in both, but the consequences at the RNA levels are less severe. And importantly, the cell compatibility, CRISPR works better on dividing cells, whether ADAR works on non-dividing cells, including neurons. At the clinical level, CRISPR is already at the clinical stage, whether RNA editing is not. So it still has a way to go before going to the clinic. Now I want to discuss the model that we're using. So Tatiana Rabinsky from our IPS core facility generated iPSCs now from 3 STXBP1 patients, but we're focusing on 2, the R406H mutant and the R292C mutant. Which doesn't or is not suitable for ADAR editing, so it's more like a negative control for us. This is just the characterization of the iPSCs. Then we decided to differentiate ourselves into induced mononeurons following an established protocol, a 30-day protocol. You can see that these neurons express, for example, the mononeuron marker CHAT, and it also expresses STXBP1 endogenously. One should also ask why mononeurons. So first of all, it induces model diversity. From what we've seen, most of the models are focused on the cellular models are focused on cortical neurons. So this shows another type of neurons. STXBP1, as we've shown, is endogenously expressed at the neuromuscular junction, which is a specific type of synapse. It might be linked to the psychomotor symptoms that are associated with STXBP1, and at least mononeuron processes are outside of the BBB, and so they could possibly be more clinically accessible. Okay, so in order to test our neurons functionally, we seated them on a microelectrode array here, and we show the 406 family and the 292 family with significant differences in some of the parameters between the patients and the controls. And if we combine them together, we have significant differences both at the number of spikes per minute and the number of bursts per minute. When we ran mass spec analysis on the all proteins, we could see that the patient lines here, marked in triangles, are separated from the control lines on the PC3 axis, which has 12% of variant genes. And if we look at the GO terms that are associated with these genes for each one of them, we can see that some of them are shared, are specific to neurons or synapses, but also that some of them are not shared. So while there are common pathways, there also might be differential protein expression for each patient that we have here. Then we looked at the DNA and the RNA levels of STXBP1. This is the 406 patient, which has the heterozygous mutation, and same go for the second patient, which is not appearing here in the control, as expected, of course. At the RNA level, we see a similar picture with this heterogeneous expression, but Sanger sequencing is not the best accepted tool for quantifying the level of peaks that we have here. So we also looked at bulk RNA-seq, and here we can see this 50% distribution at the RNA level in both of the patients. To make it more accessible for us and cost-effective, we developed an amplicon sequencing quantification, which also allows us to show this 50%, which we actually would like to change eventually with the RNA editing. Okay, so the next question we ask is whether this is haploinsufficiency, and I know that it is well known that STXBP1 is associated with haploinsufficiency, but the way we wanted to check it is using this mass spectrometry analysis. Before going into the analysis, we digested all of our proteins with lyse-C. This is according to a standard protocol, with the aim of differentiating between the wild type and the mutant allele. When we look here in green, I marked all of the peptides within STXBP1 that were detected by mass spectrometry. And here you can see the 292 position with the R. And when we look at the 406 position, we don't have coverage of this peptide, so I won't be able to drive the same conclusions. But if we focus for a second on the R292, we can see that both in the control and in the patient, there is only one protein that is detected, and it's the healthy protein. So in the patient, the situation is that the only protein, the only STXBP1 protein that is detected is the healthy allele. And if we take it with the overall quantification of STXBP1, it of course shows again that 292 is driven by haploinsufficiency. Now, if we look at the 406 line, we didn't get the same differences that we expected. And this is different from a recent paper that came out with the mouse carrying the same mutation. So I'm carrying this to the next slide. We thought that it could be because of the differentiation potential of the neurons that we obtained for these patients. So if we normalize the levels of STXBP1 to a neuronal marker, we do get this, it's not significant, but what seems to be also an haploinsufficiency case. But we also try to look specifically at this target. And for doing this, we first digested our protein extract with chymotrypsin. And here we were able only to detect in 1 of 6 samples the disease allele. So we have difficulties believing it, and we're still working on trying to do or to reach the same conclusions as we could for the 292. Okay, now back to the RNA editing. Our approach was to design guide RNA, clone them into a plasmid, a lentiviral backbone, produce lentiviruses, transduce our mononeurons or targeted cells with this plasmid, which also has the GFP. Then we can store the GFP positive cells and extract RNA and quantify the levels of each allele. So this is the transfected cells. We could sort this portion out, which is about 50% of the cells. And we first looked at the relative RNA expression of the guide. And we should compare here the untreated with the GFP positive. We see that everywhere that we transduce the cells, we do have guide that is expressed. Now what happens at the RNA level? Because this is the interesting question. This is how the untreated cells looked like. But when we use the 140 nucleotide long guide RNA, we're able to change this, to shift this heterozygosity towards the G allele, which is the healthy allele. And we did this with several guides, and we're able to reach about 6 to 7% in the 140 long guide. Now, 7% is obviously not what we're aiming for. So how can we enhance this? And this would be in future work. This paper here that came out in 2022 showed that if you use circular RNA, you augment the stability of the RNA, and therefore it stays in the cells longer, and ADAR is more effective. And later, the lab of Thorsten Stafford from University of Tübingen also used circular RNA, but he also studied the guide in the context of the nucleotides that are around the base that we try to edit. And by doing this, they're able to reach 70 to 90% editing in multiple sites. And so Thorsten is now our collaborator, and he's designed the guides that we're now cloning into the plasmids, and hopefully by the next meeting, we'll be able to show you improved RNA editing efficiency. And with that, I would like to stop by thanking everyone that is involved. I think I mentioned everyone along the way, and I'll be happy to take any questions.
I get fantastic work, thank you. What is your idea about the 7% increase? So what is holding this back, the efficiency?
So from what we think, or from what we learned from the Thorsten studies, first of all, the stability of the guide RNA, and then the context of the nucleotide, what is next to the nucleotide, could also change the efficiency of the process.
In that circularized study, how much did they increase the levels? Could they increase the levels?
So they were able to reach 70 to 90% at multiple sites.
Okay, great. Yeah.
Which is context dependent.
Okay, thanks.
Okay, if there are no more questions, Gad, thank you very much for your talk. We really appreciate it.
Thank you very much. See you next time.